and what's involved in using a gene probe. It basically involves using a strand of mRNA with a radioactive chemical bonded to it, and this is responsible for a specific gene. It's coded for a specific gene which is responsible for a certain trait in the genetic information. And then of course you've got DNA, and the whole process starts with isolating the DNA in a warm environment. The warm environment breaks the hydrogen bonds in the DNA, so therefore it strands off, and then the mRNA responsible for that gene is able to bond with it. The fact that it can identify a specific trait which could be linked to genetic diseases, genes that create hemoglobin, and then it gives us clear evidence that the genetic information that is found in all cells in the body is the same, basically, which can be useful. In red blood cells, we are able to identify the fact that production of hemoglobin is an active role in their life cycle, whereas cells in the brain still have this genetic information to create hemoglobin. It's just not actively occurring in their life cycle. That's it. So yeah, that's it, stranding off, and this is the DNA, and then you've got the mRNA with the radioactive probe bonding to it. And the reason it's got a radioactive probe is so that when it is bonded to the DNA, we are able to identify it later on, so we can show that it is responsible for a certain trait. Its other importance is that...
Questions?
No.
No questions?
No questions.
Gene probes. There isn't an actual definition, it's just strands of mRNA which with a radioactive base which bonds to the DNA and show a specific gene.
Done artificially, or is that natural?
That is done artificially.
Oh, okay.
They know the coding for
Yeah,
that
yeah.
trait. And then they just need to identify it in a specific individual.
Okay, you talked about hemoglobin. Are there any other examples you can give?
Cystic fibrosis is a genetic disease which can be identified so long as they know the correct coding for the gene. And if there is no DNA RNA hybridization occurring, then they do not have that gene which is responsible for that trait.
Okay, thank you.
Dr. Wayne, and I'm a specialist in overcoming male infertility. Now you're all here because you have questions on overcoming male infertility, and before I get on, I would like one volunteer to go ask Headmaster, but he's filming, so Harrison, if you could please come up and draw a healthy sperm cell. A little bit bigger, as if it was like a large dog. Yeah, okay, good. Just keep it nice and simple. Thank you very much. Okay, unfortunately I have to make a few corrections here. Okay, so here we have the acrosome, which has the enzymes. Here we have the nucleus, which is the genetic material. We have the mitochondria, which has all the energy for the tail, which is called the, do you know that, microtubules. I'm not going to write it down, so I'm going to put it. Microtubules, and this is the tail. This is a healthy sperm cell. The tail swims towards towards the egg. The acrosome releases its enzymes. The nucleus goes in. You all know this. I'm going to show you some pictures of some unhealthy sperm. So this is healthy. Well, it doesn't look very healthy, but it's meant to be healthy. These are some pictures of some unhealthy sperm cells. As you can see, this is healthy. So these are unhealthy sperm cells. As you can see, they all have something wrong about them. So they have maybe an extra head, or a deformed head, or an extra tail. And for obvious reasons. So if it was to have two tails, it would swim in circles and it wouldn't be able to go to the egg. If it had two heads, it would have two sets of DNA, which obviously wouldn't work. So that's the main. One of the main reasons of infertility. So quality of sperm. So abnormal sperm. There's also, if you have a low sperm count as well. That's the other reason. I don't know if I should bother writing it down. I don't know if I'm wasting time. Low sperm count. That has got many reasons. You could be, well, both of these, you could be born with them, could be genetic, or it could develop. For example, if you develop cancer, if you get infections, if you get an injury, there's a few other, if you're taking drugs, steroids, marijuana, cocaine, they all kind of come down to this. Another reason, so I'm going to draw, I'm going to draw the testes, so we've got the testes here, and that there. So you've got the testes here. I don't really know how it looks exactly, but I'm just going to draw it like this. And you've got something that goes like that. And basically, the sperm comes from here, and as it goes up, it picks up fluid from all these different glands, and then comes out of the penis. These glands is where it picks up, I just said that, it picks up the fluid. If, for example, there's many reasons, if you have cystic fibrosis, you will be missing somewhere here, you'll be missing it, and basically the sperm wouldn't be able to get to the glands and outside. There's other reasons, just general blockages, obstructive azoospermia. That's a condition in which genetically you are missing this and you just don't have the ability. There's a few other ones. There's ones where there's something happens and it kind of ends up going, instead of going out, it goes into the body. There's many other reasons to do with, just small reasons that could cause that. I've covered everything. Now we're going on to treatment. The first treatment, which is the main one, is in vitro fertilization, in which you would get a healthy egg from the female, and you will inject. I'm going to do one of Dr. Snellborough's injection pictures. You would inject the sperm. You inject the sperm straight into the egg, and then you would wait for it to turn into a zygote, and then you would place it into the uterus. Second one, for men who cannot actually produce sperm, they can. Another reason I should say is that you can produce sperm, but your sperm doesn't mature, which means when it comes out, it's not mature enough to fertilize. They can then extract them and then harvest them so they do mature, and then obviously do in vitro fertilization. They can also, there's surgery that can be done for these kind of problems. There's surgery that can be done to fix them. That's it. For treatment, that's it, I think. But obviously, you've come here and you've got a problem, but you're probably thinking, well, it would have been better if I just never got the problem in the first place. If you're unlucky enough to have it genetic, then there's obviously nothing you can do. But ways to prevent infertilization is watching your weight. So if you have a very high BMI, you are more likely to be unfertile. Your diet, fruit, vegetables, exercise. Like I said earlier, drugs, marijuana, cocaine are all very bad for your sperm count and just general healthiness. Damaged testes. So just generally, if you're playing cricket, protect your testes. Another thing is actually your testes need to be 2 degrees lower than the rest of your body for them to be made. So, for example, if you are worried about low sperm count, then you should avoid getting your testes hot. So, avoid hot baths, just general heat. Yeah, that's it. Thank you very much.
Any questions for Dr. Wing?
Yeah.
What's the name of the fluid that is made in burns? You never said the name.
Oh, semen. So, there's different glands and all comes together to create semen, which has the nutrients that the sperm needs to get to be able to swim. Any more questions? No?
How are you said that semen contains the nutrients
Yeah.
used by the sperm?
Yeah.
Where? How?
Ooh, that's a very good question, sir.
Thank you.
Well, I did kind of contradict myself because the mitochondria actually is actually where the energy comes from. The semen also helps because the sperm, I think I said that wrong, didn't I? The sperm cannot travel unless it's in some sort of liquid. So I think the semen is actually for transportation, so the sperm can swim in it. Is that right?
Yeah.
Is that right?
Yeah,
Yeah?
that's right.
Yeah, that's right. Sorry, I was
Okay, thank you, Dr. Wing.
Hello, I'm Nick Burnage, and I'm here to talk today about Wu Suk-Wang therapeutic cloning. So here's research. Wu Suk-Wang claimed to have developed sets of cells, which are stem cell lines, for individual patients. He claimed that stem cells could be differentiated into specific tissues. So he would be able to take them from totipotent, where they are undifferentiated, and make them into whatever cell he whatever cell he wanted them to become. So it could become a lung cell, a heart cell, a brain cell. So what is therapeutic cloning? Oh, back. Okay, so what is therapeutic cloning? Therapeutic cloning is a technique used to produce healthy tissues. The aim is to treat someone with faulty cells and replace them with new healthy ones. And it allows you to replace faulty organs with new healthy working ones. So say you had a lung puncture and you needed to replace a lung. You would be able to get rid of that lung and replace it with a new lung working, so you get your 2 lungs working again. So I'll now draw therapeutic cloning. And how you would be able to replace the faulty cells with new healthy ones. Right. Okay. So firstly, you get the man who's upset. He's upset because he has faulty cells, and that means he has, for example, a liver damage. Okay. So you would take, then you get a new healthy man who has a good healthy liver, and you take out any body cell, because all cells are the same, apart from your gametes, which is different. So you take his one with the nucleus in it, and you get a female and you take her egg out with the nucleus, but you get rid of this nucleus, so chuck it away. So then you're left up with an egg and that. Oh, not an egg, female. And you would put this nucleus into here. So then you have formed a pre-cell, an embryo basically. So here's the new nucleus and the cell from the female. And then you would give this a little electric shock, but a very, very small one, not really anything big. And this would make lots of new ones. You get lots of new embryonic cells. And then you would separate all these cells out, so you would get one there, one there, one there, one there. And then at this point, you are now able to make these into any tissue or organ that you want. So for example, for this man, we would need to be making a liver. So that we can now make into a liver, or we can make it into a heart, or a brain, or something. And then, once you've got that, you would then remove the faulty liver from this man and replace it with this new one. So therefore, he now has a new functioning liver. Okay. So therapeutic cloning is governed by the Human Fertilisation and Embryonic Act of 1990. A pro-life group from High Walk won a legal challenge for this to not happen because they thought it's unethical that you are making stem cells, which is basically killing life. And this meant all forms of cloning would be unregulated in the UK. But however, at the University of Newcastle, they are now able to investigate treatments for diabetes, which is the lack of insulin, Parkinson's, which is when your nerves are damaged, so you become paralysed, and Alzheimer's disease. Okay. So however, Wu Suk-Hwang's work in 2006 was discovered to have been faked. He had done nothing and lied about everything just to get his publicity up. Thank you for listening. Any questions?
So when that's still possible.
What's not possible?
It's still possible. Therapeutic cloning.
Yeah, therapeutic cloning is still possible, yeah, but all the work that he had done was a lie. So he had lied about all the work that he had actually done. However, I've made it sort of look quite simple here, but it's really not. It takes years to actually master how to do it, and no one has. No. Yeah.
How did he think he was going to get away with it?
I don't know. He was trying to just raise his publicity, and then I don't know. I don't think he was ever going to get away with it because other scientists are going to try and do the work that he did. And
They see you're not very clever.
he wasn't very clever. Maybe not. Haven't done it.
You've got those undifferentiated cells there, and then
Yeah.
they miraculously turn into a liver or heart. How do they
Because
turn?
you were able to, well, with this little electric shock that you give them, you are able to make them into what you want. So change them into whatever cell, organ,
So
tissue
what
you want.
differentiates them into another cell?
I don't really know. But that's why it's so hard for anyone to actually do, because no one really knows how to turn it into a liver, heart, or brain cell.
Well,
organ.
You put it in certain areas of the body and prompt it in order to change.
Extension work, I think.
Maybe.
Thank you, Dr. Burnage.
This is Dimitri, and
Hello,
he's a half
Dimitri.
boy. He's 17 years old, but in 2003, he got shot in the heart. He didn't die, but he got a massive heart attack. Like loads of muscle tissues were destroyed. So he went to the hospital straight away, and obviously he needed a heart transplant. He needed a heart transplant, but they couldn't find anything, like, because you need really quickly, and you can't find like a heart like this. So because the hospital was starting a program of research of adult stem cells, they decided to start to try it on him. So he was like the first guy ever to be treated with adult stem cells. So what they did was they gave him drugs to produce new stem cells in his blood, and then they harvested them and transplanted them into the coronary artery, which then carried them to the damaged heart muscles. And his heart had like increased, like before when it was damaged, it was only pumping, it was only pumping 25% of the blood, when normally it should be 55 to 65%. And then after they used the adult stem cells, it started to pump 35%. So, which is enough for him to go back home and have normal life. And that's it. So he survived.
Questions, please?
What drugs did he take? Do you know?
Drugs.
Yeah.
Where is he from?
I don't know. Didn't say in the book.
Dimitri.
I mean, like, what country was he from?
No, it didn't say anything.
How extensive was the damage?
It was really bad.
And how do they transplant the stem cells into the coronary artery?
Big injection.
Probably injection, yeah.
And why didn't the stem cells flow through the heart?
Flow.
What made them stop there?
Like, because it was damaged? No.
Maybe? Okay. Any other questions?
What's your opinion on stem cell research?
I don't know, I think sometimes it's good, but other times, like, it depends, like, the adult stem cells not always work.
No, I mean like ethical issues.
And like, if it's embryonic, it's bad, because it's like you're killing baby or something, but then the other way, it's helping people as well, so I don't know.
Interesting take on it.
Thank you.
So, there's one for you, sir. So, is everybody ready?
Are you ready?
Yes, I hope so. Alright, my topic is on mysis, I think. Yeah, mysis. And as you can see from the diagrams that I have given out to you, meiosis is where one cell, or one little cell, splits into several different cells with all its genetic material inside of it being split and obviously, reproducing and going to the different cells. Now, in the first phase, this interphase, where there's 46 chromosomes in each cell normally, and that's how it is. And then there's prophase 1, where each chromosome, doubles like that and replicates. So each chromosome turns from 46 chromosomes into 92 chromosomes in each one, and then they go into the homologous pairs, which is that's pretty much the 96 chromosomes, and obviously you have the chromosomes from mum and dad and parents, and then there's crossover happens in the prophase 1, where a certain section from each the chromosomes switch over, switch places, which helps for variation and mutations. Obviously, if you provide variation in the genes, that can be transferred into the gametes, which will produce a zygote. And then there's the next stage is the metaphase 1, where all of the all of the chromosomes line up in a row as such, then they align in the middle of the cell, and well, in the metaphase plate, as it's called, and then the spindle, lines up on each side, as on this side as well, and getting ready to pull all of the cells apart, but not quite yet. And spindle is the fibers that are made up of microtubulars and the protein tubulin and other motor proteins as well. So that's what helps it split up. And then there's telophase, anaphase one, the centromers, which is this little centre bit keeping all the chromosomes, the two parts together, and the centromere do not divide in it, but they still hold in place. But what happens is that each pair separates as such, so, and then getting ready to divide pretty much, which is where telophase 1 comes in. And telophase 1 is where the nuclear membrane reforms to the cells, reforms and the cells begin to divide. In some cells, this continues to full cytokinesis, which I'll get onto later, and there may be a period of brief, prolonged interphase. During this interphase, there's no more further replication of the DNA. And then there's metaphase 2, which new spindle is formed, and the chromosomes are still made up of pairs of chromatids, and chromatids, that's one chromatid and that's another chromatid, so two chromatids. Where the chromatids line up on the metaphase plate again to get split up. And in phase anaphase 2, the centromes divide in this phase now, and the chromatids move to the opposite ends of the cell, again to split. In telophase 2, nuclear envelopes reform the chromosomes and return to their interphase state. And then there's cytokinesis, cytokinesis, which is where one cell, with all of the chromosomes, splits into two, and then cytokinesis is it was 4 daughter cells with half the chromosomes in the original diploid, meaning half the 96 chromosomes. So I have 92. So I have 46 chromosomes in it. And yes. So any questions?
What's the difference between mitosis and meiosis?
Well, obviously, meiosis is where gametes, obviously, parent cells form and then it reproduces and replicates, whereas mitosis is single cell, asexual reproduction.
Can you tell us anything about abnormal splitting of chromatids, such as Klinefelter's syndrome or Turner's syndrome or Down syndrome? How might that happen in your diagram?
I do not know.
Anybody know about what happens with Down's
It would
syndrome?
be an extra set of chromosomes.
Extra set?
Well, usually one of the spindle fibers breaks and then the chromosome goes to the other side.
So 2 sets of those daughter cells will have one more chromosome. So they won't have 46, they'll have 27. Extra.
Wait, one more?